Okay, so part 3, we're almost done. So the tail end of this chapter, got some more slides left to go. We're going to talk about the endomembrane system and proteins, the cytoskeleton, and then some cellular junctions. So let's start off with the endomembrane system. So inside of our cells, eukaryotic cells, we have a lot of membranous structures. And those membranous structures are kind of continuous with each other, or vesicles can kind of, pockets, membrane-bound compartments, can kind of come off of one structure, travel a little bit, and then fuse and become part of another structure. So it's this continuous membranous structure throughout the cell. Let's look here in this figure. We've got our nucleus, which you know is a double membrane structure, right, in purple. And then that's going to be continuous with this blue membranous structure called the endoplasmic reticulum. In this case, this is the rough endoplasmic reticulum. All these red dots all over it are all ribosomes. So obviously this is important in protein synthesis. And if we look here, there can be these vesicles that kind of bud off of the endoplasmic reticulum. They'll travel, and they're going to fuse with this structure here, which is called this peach structure. Again, this is a membranous structure. It's just a bunch of membrane-bound sacs. Okay, and a lot of good stuff happens in here. We'll talk about that. And then vesicles from this compartment can then bud off and travel and fuse with the plasma membrane. So when we say the endomembrane system, it's the internal system of membranes that are all kind of connected to each other, that can fuse with each other. Okay, pretty neat. So again, it's a group of membranes and organelles that work together to synthesize or make, modify, package, and transport things like lipids and proteins. Included in this endomembrane system is the nuclear envelope, lysosomes, vesicles, the endoplasmic reticulum, which we call ER for short, the Golgi apparatus, and the plasma membrane. Okay, the endomembrane system is involved in taking stuff into the cell, which is called endocytosis, as well as getting stuff out of the cell, excretion or exocytosis. The brain system plays a key role in that. Okay, so we've already talked about the nucleus and the nuclear envelope. Let's go to the next part of the endomembrane system, which again is continuous with that nuclear envelope, the ER, or endoplasmic reticulum. Again, this is a series of interconnected membranous or membrane-bound, hollowed out sacs and tubules, and it is continuous with the nuclear envelope. What happens inside of these membrane-bound sacs? Well, we actually have two types of endoplasmic reticulum. We have the rough ER, that's shown here, and it's rough because you see those little red dots spotting it. It has a rough surface. It's a bumpy surface. Those little red spots are all ribosomes. And then we have the smooth ER, which you can see is everything over here. And the smooth ER does not have ribosomes, smooth surface. Okay, but both the rough and the smooth ER are membranous structures. And the inside, which we can see, a lot of this has been kind of cut, so we can see inside here, inside here, inside here, this whole opening here. There's an internal space inside of the endoplasmic reticulum. So what's happening in there? It depends on if we're looking at the smooth or the rough ER. So the endoplasmic reticulum that's studded with ribosomes, the rough ER, proteins will be made on those ribosomes, and they'll be inserted either into the membrane of the rough ER or right into the lumen, the space inside the ER. And those proteins are going to be modified by the endoplasmic reticulum. The endoplasmic reticulum, the rough endoplasmic reticulum, is involved in protein modification. We can get a little bit deeper into that in just a second. The smooth ER is involved in things like lipid, carbohydrate, and steroid synthesis, as well as detoxification of any medications or poisons, and calcium storage, okay, which is going to be really important in certain cells like muscle cells. Okay, again, it's important to remember that these membranes here are continuous with the plasma, the nuclear membrane, the nuclear envelope. Okay. Okay. let me actually talk a little bit more about the rough ER. I think that there's a slide in a few minutes that shows this in a little bit more detail, but just so that you know. What types of protein modifications? Well, maybe a protein gets cleaved or cut, or maybe it gets folded in a certain way or unfolded and then refolded. Okay, so proteins can become modified in there. Okay, so the next spot that we're going to go to is the Golgi apparatus. And the Golgi apparatus is really connected to the endoplasmic reticulum. The Golgi apparatus is going to receive cargo from the endoplasmic reticulum, like maybe it's going to receive proteins from the rough endoplasmic reticulum, or maybe it will receive carbohydrates or lipids from the smooth endoplasmic reticulum. Okay, that cargo is going to come from the endoplasmic reticulum packaged into these little things called, oops, I didn't have my pen. Hold on. Okay, so again, proteins, lipids, carbohydrates, those are made in the endoplasmic reticulum, and they're going to be transported to the Golgi. So do you see this little vesicle? This would be coming from the endoplasmic reticulum filled with whatever cargo it is, proteins, lipids, carbohydrates. It'll travel through the cytoplasm, and then it will fuse with the Golgi. And the Golgi actually has two faces or two sides, a cis face, which is going to accept vesicles from the endoplasmic reticulum, and a trans face, which is going to export vesicles carrying cargo. Okay? The job of the Golgi, once it gets stuff from the endoplasmic reticulum, is to take that stuff and to sort it, to package it, to modify it further, and to get it to where it's supposed to be. So it kind of tags it. I always think of the Golgi as kind of the mailroom. It's the packaging center. You know, the mailroom receives a package, it makes sure it gets to where it needs to go. And that's what the Golgi does. It receives packages from the endoplasmic reticulum, and it makes sure those packages end up at their final destination. Okay? The Golgi kind of looks like the endoplasmic reticulum. It's a series of membranous sacs. Kind of easy to find in the cell because it always takes on the same shape. There's a series of flattened sacs that looks pretty much like what we're seeing here in this figure. Again, there's a cis and a trans face. The cis face is the receiving end. The trans face is going to be the export side. And when these vesicles get exported, it depends on where the contents is destined. They might travel to the plasma membrane and then fuse with the plasma membrane, or travel to some other organelle in the cell and fuse with that. Okay? So the Golgi apparatus is the sorting center of the cell. We're going to come back to lysosomes again. I told you we would. So again, the lysosome I told you to think of as the garbage disposal. And that's what they are, but they're part of the endomembrane system. So they will receive or fuse with vesicles either carrying old, kind of used up organelles, or they might fuse with vesicles carrying stuff that's been ingested by the cell. It's been taken up through endocytosis or phagocytosis. So that's what's being shown here in the picture. This is pretty neat. So this is a cell probably of the immune system. It looks just like a white blood cell called a macrophage. And here is a food particle, okay, shown here, or it could be, if it is an immune cell, it could be a bacteria, something like that. What's going to happen is the plasma membrane is going to engulf this in a process called phagocytosis. Okay, once inside, that food will be in a vacuole, right, made of the plasma membrane, made from the plasma membrane. That will fuse with the lysosome. And the structure here will be called the phagolysosome. Those enzymes from the lysosome then become activated. They're going to chew up that food, and then that food might be recycled, by your cell, or it might be just spit right back out. So that's called exocytosis. So that that vesicle carrying all that chewed up food will travel to and fuse with the plasma membrane to dump out the contents. Okay, so see how the lysosome is part of that endomembrane system? It can fuse with vesicles and vacuoles. It can fuse with the plasma membrane. Okay, so that's it for the endomembrane system. Again, a series of membranes found within the cell that can fuse with each other to become part of each other. It includes the endoplasmic reticulum, both rough and smooth, the Golgi, vesicles and vacuoles, lysosomes, and the plasma membrane. Next up is the cytoskeleton. And when you think of the cytoskeleton, I want you to think internal skeleton or internal network of protein fibers that help to maintain cell shape, just like your skeleton helps to maintain your body shape. Okay? Helps to hold you up, it's structural support. It's the same idea here with the cytoskeleton. So again, it's a network of protein fibers, and its job is to help maintain cell shape, help to secure organelles in place, and to allow the cytoplasm as well as vesicles to move. Some elements of the cytoskeleton are also involved in whole cell movements or gross cell movements. Okay, we'll talk about those. There's three main types of protein fibers involved here: microfilaments, intermediate filaments, and microtubules. And I'm just going to introduce you to each of those and give you some examples of where they're used in the cell. So the first are microfilaments. And if you compare their name, to the other names, micro meaning small, these are going to be the narrowest. Okay? In fact, their diameter is only 7 nanometers, which is really, really small. Microfilaments are made up of 2 intertwined strands of proteins called actin. Another name for microfilaments is actin filaments. So if we look here on our figure, we can see that there's 1, 2 intertwined. You can see how they kind of wind around each other, filaments. And each one of these green spheres represents an actin protein. So it's a strand of those. Okay, what are these used for? What do they do in the cell? They're involved in cellular movement. With the use of energy, or ATP, and with the help of a second protein called myosin, they're going to help stuff to move within the cell. What kind of movement? Well, cell division. So when we talk about mitosis or meiosis in animal cells in a few chapters, we're going to be saying that in animal cells, a structure called the cleavage furrow forms. This happens at the very end of cell division. And it's actin, or microfilaments, that are involved in actually pulling the membrane and pinching the two cells apart. So these are involved in cell division. They're also involved in cytoplasmic streaming, which is literally kind of the stirring up of the cytoplasm. They get the cytoplasm moving. This is really important in cells that are kind of stationary, because you want the cytoplasm, the contents, the nutrients, the waste in the cytoplasm to be kind of evenly distributed throughout the cytoplasm. So that's cytoplasmic streaming. Muscle contractions. Microfilaments are involved in the shortening and lengthening of muscle fibers. And the process that we actually just saw 2 slides ago, this is called phagocytosis. We said that the membrane of this cell kind of wraps itself around. It's actually actin filaments that push out the membrane and allow that engulfment to occur. The pushing out of that membrane they're called pseudopodia. Okay. Next are the intermediate filaments. And as the name implies, these are going to have an intermediate diameter, size. Okay. These are going to be made up of several strands of fibrous proteins kind of wound around each other. They're about 8 to 10 nanometers in diameter, so slightly larger diameter than we saw with the microfilaments. And they're going to be mainly functioning in structural support. They help to maintain cell shape, they are tension bearing, and they also help to anchor organelles like the nucleus in place. So the nucleus isn't just kind of floating aimlessly around in that cytoplasm, as well as other organelles. Okay, last one. microtubules. These are going to be the largest of the bunch. Microtubules are hollowed out tubes made up of alpha and beta tubulin. Those are proteins. And the diameter of a microtubule is 25 nanometers. So again, these are going to have the largest diameter. They've got a variety of different functions. They're involved in maintaining cell shape. They can resist compression. They also provide a track for vesicles to move. So remember we said that vesicles can move from the ER to the Golgi, or from the Golgi to the plasma membrane, or from the plasma membrane to the lysosome. When those vesicles are moving in the cytoplasm in between compartments, they're not just moving again aimlessly around the cell. They have a clear path, and that path, or literally kind of like a road, is created by these microtubules. So a vesicle can move from the ER to the Golgi by literally kind of walking along the microtubule. There's proteins and energy involved in that process, but that vesicle literally gets carried along that road. It's pretty cool. What else? These guys are involved in separating chromosomes during cell division. So we just talked about centrioles at the center of the centrosome, and we said that the microtubules, these, grow out from there, attach to chromosomes, and then pull the chromosomes back. So that's the job of these, of microtubules. They're able to do this because they can form and break down quickly. So you can take and you can add subunits to make these grow longer, and then you can take off subunits to make them become shorter. Okay, so these will grow out during mitosis and meiosis to attach on to chromosomes, and then they get shortened from this end, and those chromosomes will get pulled in that direction as they get shortened. Okay? Okay, so again, I just wanted to give you a little bit of information of tubules. Hold on a second here. Yeah, one of the other major roles of microtubules, besides being used in cell division, is that they're involved in movement of cells. So when we look at structures like flagella, whip-like tails, or cilia, those are going to be structures, again, that are involved in movement. Flagella, we saw when we looked at the bacterial cell. Another example of a cell that has a flagella would be like a sperm cell. Cilia we saw as well earlier in the chapter when we took a look at the paramecium. It was covered in that hair-like substance. That's cilia. Both flagella and cilia are going to be involved in movement of cells. Okay. Flagella, again, long, hair-like, whip-like, tail-like structures. They are extending from the plasma membrane. They're going to be used to move the entire cell. Usually there's only one or two flagella per cell. If we look inside of a flagella, we find microtubules. Those are going to be the structures that are going to allow that flagella to move like a whip-like tail. Cilia, short, hair-like structures that are usually found on the surface of a cell. There's usually a ton of them. Again, if we look on the inside, we're going to find microtubules. And those microtubules are going to allow those cilia to move back and forth. Okay, which they may use for movement, but more than likely they use to move substances across the outer surface of their cells. Okay, this is just showing the inside of a flagella or cilia. And in either case, we find the same thing. So we find a ring of microtubules. In this ring, there would be 9 doublets. Okay, so 9 structures of 2 microtubules. And then in the center, we find a single doublet. So 2 microtubules. Whether we're looking at cilia or flagella, we find the same structural arrangement. It's pretty neat. Okay, so pause here and complete these Are You With Me's, and then come back. I have a couple questions for you. Okay, so first question. Which of the following is not a component of the endomembrane system? The mitochondria, the Golgi, the ER, or the lysosome? Which is not part of the endomembrane system? So hopefully you answered red, the mitochondrion. It's not part of the endomembrane system. It's a double membrane bound organelle, but not part of the endomembrane system. Next question. The process by which a cell engulfs a foreign particle is known as, you had to pay attention on this slide, is that known as endosymbiosis, phagocytosis, hydrolysis, or membrane synthesis? Okay, so let me take you back to that slide. Make sure we got it. I mentioned the endosymbiont theory, but that's not what we're talking about here. We're talking about this. So in this figure right here, this is called phagocytosis. It's the engulfing of food or some other particle by the membrane. Okay, so here, question number 2, the answer will be blue, phagocytosis. Next question. Which of the following is most likely to have the greatest concentration of smooth endoplasmic reticulum? A cell that secretes enzymes, a cell that destroys pathogens, a cell that makes steroid hormones, or a cell that engages in photosynthesis? So we got to go back here. I'll clue you in. So the endoplasmic reticulum, and we said that the smooth ER is responsible for lipid, carbohydrate, steroid synthesis, detoxification, and calcium storage. Okay, so hopefully you answered green. It would be a cell that makes steroid hormones. Okay, which of the following sequences correctly lists the order of the steps involved in the incorporation of a proteinaceous molecule within a cell? Oof, that's a lot of words. Okay, so which sequence is the steps? So let's look here. So in pink, protein synthesis of a protein on the ribosome, modification in the Golgi, packaging in the endoplasmic reticulum, tagging in the vesicle. So is that the order? Or blue, synthesis of the protein on the lysosome, tagging in the Golgi, packaging in the vesicle, distribution in the endoplasmic reticulum. Does that make sense? Or green, synthesis of a protein on the ribosome, modification in the ER, tagging in the Golgi, distribution via the vesicle. Does that make sense? Or yellow, synthesis of the protein on the lysosome, packaging in the vesicle, distribution via the Golgi, tagging in the ER. Okay, so immediately you should eliminate blue and yellow because both of those say synthesis of protein on the lysosome, and we know that that's not where proteins are made. Proteins are made on ribosomes. So blue and green are still good. So where does that protein go next? Does it go into the Golgi to get modified, or does it go into the endoplasmic reticulum to get modified? It's going to go into the ER. Okay, and then from the ER it goes to the Golgi, and then from the Golgi it goes to wherever it's destined to be. Okay, so green would be the correct sequence of events. Next question. Which of the following have the ability to disassemble and reform quickly? Microfilaments and intermediate filaments, microfilaments and microtubules, intermediate filaments and microtubules, or only intermediate filaments? So I'm not sure if I addressed this as well as I should have, so let's go back for a second. We know that microtubules can disassemble quickly, okay? Let's take a look at these intermediate filaments. These are made of strands of proteins wound together, and let's compare those to intermediate filaments that are made up of these individual proteins. Microfilaments and microtubules are both made up of protein subunits, and both of them can be formed and taken apart pretty quickly. Whereas intermediate filaments are made up of these strands of proteins, not subunits, and so these cannot be taken apart quickly. So let's go back to our Are You With Me? Okay, and so the answer here would be blue, microfilaments and microtubules. Anything made up of small subunits tends to be able to form and be taken apart pretty quickly. Made up of large protein filaments, that would be more difficult. So intermediate filaments would not be formed or taken apart quickly. Okay, last section of the chapter. Thank goodness, this is a long one. So connections between cells. So we know that in multicellular organisms, cells will come together to form tissues. Okay, and in tissues, these cells sometimes are very close together, sometimes spaced out a little bit, but they need to be able to communicate with each other. In animals, we have something called the extracellular matrix that will be formed by cells outside to the exterior of the cell, and it'll be composed of both proteins and carbohydrates. So that's what's being shown here in this picture. This is the inside of the cell, or the cytoplasm, and this is the outside of the cell, the extracellular matrix. And so you can see we have collagen fibers, and we have polysaccharides, and we have proteins and carbohydrates. Those are all parts of that extracellular matrix found outside of the cell. The cell actually secretes these proteins and carbohydrates. Okay, connections between cells. These are called intracellular junctions. They are direct contact between cells. In plants, we have structures called plasmodesmata. These are going to be connections in between two neighboring plant cells. So here in this figure, we have plant cell number 1 and plant cell number 2. In green, we have the cell wall. Okay? In yellow, we have the cytoplasm, and where I drew my numbers actually represents that big central vacuole. The plasmodesmata is shown right here. It's a direct connection between cell 1 and cell 2. Directly through the cell wall, the cytoplasms are connected to each other. This allows materials to be passed from one cell to the next without having to use energy to bring that material out and then back in, through a cell wall, through the other plasma membrane. Instead, here we have a direct connection. So that's between plant cells. These are channels. Between animal cells, it's different. We've got a couple of different types of junctions. The first type of junction here is called a tight junction. We're going to find this between cells that are butted up right next to each other, and in regions of the body where we need a watertight seal. Okay, these tight junctions, it's kind of like the two cells, their membranes have been sewn together, and there's really not a lot of space between those cells, so that nothing can pass through. It prevents leaking or anything really from getting in between those cells. We find this when we're looking at epithelial barriers and linings of internal organs and cavities. We don't want fluid, leaking into or out of those types of structures. Okay, so again, those are called tight junctions. Two cells that are literally sewn together. Their plasma membranes are sewn together. Proteins will accomplish that. The second type of junction that we find in animal cells are what are called desmosomes. I like to think of desmosomes like snaps. Instead of being sewn together or zipped together, these are just snaps, okay, that hold two neighboring cells together. They're not waterproof. Instead, materials can get between them, okay? These are important in regions of the body where the tissue needs to be able to stretch. So if we're talking about your skin or your heart, right, that's an organ that has to be able to stretch. The lining of that organ is made up of cells that will have these types of connections in between them. Or muscle cells, if we're talking about cardiac muscle cells or skeletal muscle cells or smooth muscle cells, any of those will be connected to each other, but again, those connections are going to allow flexibility or stretching of that tissue. Those are called desmosomes. I've got one other type of junction to cover, and these are called gap junctions. So gap junctions are going to be the equivalent of plasmodesmata, but we're talking about animal cells here, right? So we said that plasmodesmata were channels connecting two neighboring plant cells. So in animal cells, we have gap junctions that do the same thing. So we have two cells right next to each other that are going to be directly connected. Their cytoplasms will be directly connected by these channels called gap junctions. So here we go. In this figure, we've got two cells. So we'll say this is cell 1 and cell 2. And so here's plasma membrane 1 and plasma membrane 2. And again, in between them or connecting them are those gap junctions. They're going to be composed of proteins, and they can actually either be opened or closed. And they're going to allow communication, passaging of molecules between those two cells. Again, that's going to save energy for those cells. Okay, your turn. So again, pause here, finish this last "Are you with me?" And then I have one more set of questions for you. So come back when you're finished. Okay, so just two questions here. First question, which of the following are only in plant cells? Do we have gap junctions only in plant cells, desmosomes, plasmodesmata, or tight junctions? Which one of these is only found in plant cells? So hopefully you answered in green here, plasmodesmata. Those are going to be specific to plant cells. Everything else here is specific to animal cells. Okay, last question. Diseased animal cells may produce molecules that activate death cascades to kill the cells in a controlled manner. Why would neighboring healthy cells also die? Okay, this is a good application question. So in pink, the death molecule is passed through desmosomes. The death molecule is passed through plasmodesmata. The death molecule disrupts the extracellular matrix, or yellow. The death molecule passes through gap junctions. So you've got a couple of cells here. So you got cell 1, let's say, and cell 2. And cell 1, something happened that's bad, and so it's going to go through kind of a programmed cell death. So we have this molecule called the death molecule that's going to be produced, and this cell is going to die. Why would its neighbor also die? What could happen? So hopefully you answered yellow. That death molecule could make it through a gap junction if that gap junction is opened, and therefore neighboring cells could die as a result of that. Okay, so that is the last question. So you can go ahead, get that "Are you with me?" posted to Blackboard, study, and then go ahead and take the quiz for the week.